So starting off here, Dan Gincel is the, I'm going to read this because this is a little bit more complicated, I think. He's the Vice President for University Partnerships at the Maryland Technology Development Corporation, TEDCO, and Executive Director of the Maryland Stem Cell Research Fund. Comes from a background in research, and also just learned that he does applications for phones, which on his own hobby time, which is pretty unique actually. So welcome to Dan. To his left is Elona Baum, who I think a lot of folks in the audience know. She's the General Counsel and Vice President of Business Development for the California Institute for Regenerative Medicine, or CIRM. Trained attorney and spent a number of years at Genentech, some as Associate General Counsel, so brings a very unique perspective, I think, to this discussion. And then to her left, Reeve, who is the Executive Director of the Harvard Stem Cell Institute, so this is kind of a home game for him. And it brings with him a lot of experience in the commercial sector with experience in management consulting and operations in technology-based companies. So we have a very, I think, a background that's also diverse in terms of not only organization representation, but experience set. So what I'm going to do is turn it over to them, and they can spend 3 to 5 minutes introducing their organizations to the audience. So, Dan?
Good morning, everybody, and thank you for the organizers for the opportunity. As I was introduced, I'm part of TEDCO. TEDCO is the Technology Development Corporation for the state of Maryland. And I think this puts a bit of a different perspective on our unique funding opportunities that we have to fund stem cell in the state of Maryland. We are looking at the technology, we're also looking at the economic development for the state, and not just moving science forward. We are focusing mostly on collaboration between academic institutes and the industry. I was actually very impressed to see a number of, actually the majority of the exhibits outside are Maryland-based companies. So this is very encouraging, and we're trying to do everything that we can to fund more and more companies. I'm going to touch base about it a bit later, but I put some of the information about our programs on the tables in front of you, and we'll talk about them in the questions.
So Dan is the one who put all the money on the table, so he's a good guy to know. And I know George spoke earlier, he has a lot of money, but he didn't put it on the table, so I think it's nice that Dan actually did. So.
Thank you, and I'd like to thank Terrapin for inviting me to speak today as well. I'm with the California Institute for Regenerative Medicine, CIRM, for those of you who haven't heard of us yet. We are the $3 billion funding agency for stem cell regenerative medicine in California, and we were created by the taxpayers of California through the initiative process, which I think some of you are familiar with. We were started in 2004, and then we went right into litigation, challenging the proposition. It seems to happen a lot in California. So we didn't really start funding the field until 2006, and we've been going strong ever since. So what do we fund? I'm sure that's the question of the day. I think we define stem cell research a little different than many define it in here. We will actually fund not only cell-related research, but also small molecules, antibodies, if they have an endogenous impact on stem cells, or in the cancer stem cell arena. So I think you've seen a little bit about that today. In addition, of course, tried and true is a pluripotent area that we fund and is a primary focus of our funding. $3 billion, as I said, which at a minimum has to be spread over 10 years. It could be longer. Our projections at the rate that we're spending have been that we'll probably have our last RFA sometime late 2016, 17, but who knows, because we only fund projects that meet a certain scientific score and level, and so we might not fund a particular round if the grants working group review doesn't feel that the projects are mature enough or warranted. And we fund across the whole pipeline, so we have a lot of monies dedicated to basic biology. Then we have an early translation round where you're just trying to find targets. And most recently, we've been funding and focusing on later stages where we're funding the pre-IND work, phase 1, phase 2, What we will not fund is phase 3. That's a policy decision. It's not a requirement of the statutes. But we figure that we're mainly here to seed, and in order to do that, we want to make sure that we spread our funding as broad as possible, so funding phase 3s makes it a little difficult to do that. We spent a lot of time building 12 state-of-the-art stem cell research facilities throughout California that will serve California and the field for years to come, even when our funding ceases. That's answering one of the questions I know that will come up. We also do some broad programs, like we're creating an IPS stem cell bank. We have a genomics initiative where we're trying to create, or will create, centers of excellence in genomics. But if we had to, and I'll stop soon, I don't want to hog the floor right now, but if we had to focus on what our 2 missions are, I would say, first and foremost, it's really to accelerate and drive transformative medicines to patients. I think that's what the Californians have in mind. But we also have an economic development focus. There's a verbiage in the statute that discusses building an economic engine for California. So we're mindful of that as well. And I'm sure we'll get more in depth as questions arise.
And thank you also for the opportunity to join you here today, even though all I had to do was go an extra 2 blocks this morning. The Harvard Stem Cell Institute was formed back when CIRM was formed. The good news is we didn't have to go through 2 years of litigation. The bad news is we didn't have $3 billion at our disposal. We're sort of a hybrid animal, and we're sort of a science experiment, a business model experiment, and maybe even a social experiment. Because the Institute was formed as a virtual organization, part of Harvard, not a separate nonprofit, but dependent upon private philanthropy, to fund scientific work across Harvard and all the Harvard-affiliated hospitals. And the reason I mentioned it's a business model experiment, each of the Harvard hospitals and Harvard are legally and financially separate institutions. So this was an organization that cut across all those organizations from a stem cell and regenerative medicine perspective. When we say, and I'll mention sort of what our agenda is, but to pick up on one of Elona's points, when we say regenerative medicine, we're thinking not only cell therapy, but we're also looking at stem cells to find drugs. So we have a therapeutic screening center. We're also looking at how do we use drugs, small and large molecules, to stimulate endogenous repair, regeneration and repair as well, in addition to cell therapy. The advantage of pulling together the university and the hospitals is that we can go from bench to bedside without having to own all the assets. So what we have done is formed an organization that now comprises 90 principal faculty, another 170 affiliate faculty across that network. We raise about 15 to 20 million a year in private philanthropy, and we spend that a year. So like any startup company, we're chasing our tail, raising money. But the good news is we don't have to go to the capital markets, but it's a different form of angel investing if you think about the previous talk, one of the previous talks. Our motivation is to do basic science, but with a disease orientation. So we're organized in terms of disease programs. We have some core facilities that help support that and accelerate the research. But we're also funding very early stage risky projects with the hope, with the idea that then people can go to the NIH, can go to other sources. So our funding is not trying to displace NIH funding. We're trying to build on top of NIH, Howard Hughes, other funding sources, provide sort of key capital, and then partner with disease foundations and companies to to leverage our capital even further to accelerate that whole process. And having both basic science people, multiple disciplines, whether chemists, biologists, material engineers, et cetera, able to work together across that network and to go from basic research all the way into the clinic is sort of a critical aspect of what we can do. Now, admittedly, that's under one organization, but we're also part of this sort of the Boston ecosystem of life sciences, and then also collaborate on the outside world. But again, it's a slightly different model, but a variation on some of the themes that you've already heard about today.
Great. So I'm actually going to join these guys up here, make it a cozy little panel session here. So I'm going to start off with a couple questions, and I'm going to give them a kind of a softball question to start off with. The real question, and it's been touched on a couple times, is what are the hurdles to funding in regenerative medicine, and do you see those hurdles being unique to regenerative medicine, or do you see those being kind of typical of life science overall, biotech overall? Start, Dan. Yeah.
Okay, I'll take a stab at this. I think the hurdles really is because, at least when we fund projects, we are looking first at the science. And I think the field is very young. The science is not yet developed enough to actually support companies that can be self-sustaining. It's a lot of exploratory scientific projects, and this is what we at least focus on in Maryland. We don't have $3 billion. On the other hand, we're not as big as California, but we focus on small exploratory projects with innovative ideas that hopefully our funding can be leveraged at a later stage. And again, going towards the regenerative medicine, we're really trying to force the academic institute who are really thinking outside the box to collaborate with companies, to bring the companies to the table very, very early. We think that's going to give a more commercial angle to the product that they're going to develop, and not just the basic science and discoveries that they do so well.
From my perspective, I think that one of the most important aspects is that what we fund in terms of translational research ends up not sitting on a shelf in 5-10 years, but actually gets connected to future follow-on financing. So what I'm trying to do when I wear the hat of BD development is to make sure that I'm letting the VCs out there know what's coming through the pipeline, letting the pharma know what's down the road, letting them know, and I think some of them are starting to bite, that, and we know some of them already have, Baxter, for example, that this is a new field. They have a chance to get in early, or some might actually feel that if they don't, they might miss the boat. And I think there's an attraction there for them to at least take my meetings. That's a good sign. And I think they're starting to realize that this is a maturing field and that they are interested in knowing more about it. I think, though, that because it's maturing, not matured, and it's therefore more risky, governments, foundations have to play a fundamental role in de-risking the financing. So when we are trying to create a vehicle for this follow-on financing, what we say is companies, academics, in one of our programs, it's not all of them, if you can have a venture with you, if you can have a pharma deal with you, you can come into this other program. We call it the Strategic Partnership Funding Program. It's a revolving program every 6 months, so it's smaller, it's more nimble. You can come through there, and CIRM will co-fund with you, and therefore help the venture and the pharma de-risk the projects that they're interested in. And I think that's a model that we'll see more and more of, because I don't know if they're going to go in, head first in a significant way without some financial de-risking as part of the equation.
Let me sort of answer that question from two perspectives. One is in the general funding arena that we deal with, the NIH is a key player in the background. And one of the issues that we're finding is that with the NIH budget having been flat over the last half a dozen years, which means it's gone down in real terms, that our scientists, the academic scientists, are spending more and more of their time writing grants and looking for money sources. But that's the general backdrop of the funding challenge that basic science has. So when pay lines are at 10% and people talk about spending 30+% of their time writing grants, that's time they're not spending in the lab. They may be time thinking about the experiments, which is a useful thing to do, but it means time they're looking for money and not actually doing the work, which means then they turn to other sources and look for other sources. On the if you look at the sort of the cell therapy part of regenerative medicine, I think one of the funding challenges is, in particular, companies and venture capitalists and others, you'll see increased hesitation about, do I fund this? or not do I get engaged? And that is the clinical trial process is less well known than it is for small and large molecules. Just one of the issues that Chris Mason pointed to earlier was, the body metabolizes, processes a small molecule a lot different than you do a cell when you're wondering, where's it going in the body? Is it staying there? Is it turning cancers? All these things that the FDA, any regulatory body wants to know. But it's harder to understand and show and prove. And so, by definition, the process of bringing something to market is going to be longer and less well known and potentially more expensive than than it is for some of the other approaches.
So all of you just mentioned that the need to interact with commercial companies to help bring research through, to interact. What specific mechanisms do you all have in place for that interaction to occur or to attract their participation in those efforts?
Well, thank you for that question. So the first thing that we have is actually in 2 weeks we have our own Maryland Stem Cell Research Symposium. It's going to take place in 2 weeks. Also a little bit of a postcard on some of the tables, and if you want more information you can come and talk to me. We bring together about 350, 400 scientists with all the companies that we have in Maryland, and we make sure that they're going to interact and meet each other and discuss future and potential collaboration. The other mechanism is really emphasizing in the RFA process that companies and academic institutes will get priority if they collaborate. And that priority, it first needs to meet some standard of scientific level that's going to be reviewed, but then they're going to be pushed up if they have this collaboration, if they have the translational potential section of that application much more defined in a very precise way. So we're using those mechanisms to first introduce them together and then ask them to submit the proposals together.
So Dan, just a quick follow-up. You're saying that proposals that actually have both involved are seen more positively than if it's a single type of collaboration or single instance?
Absolutely.
I briefly mentioned our strategic partnership funding program, and effectively that has been industry only because you either had to have raised $15 million and had a year in cash to qualify, or you had to be, for instance, an academic that had a large biopharma partner. So in effect, that's our industry only RFA, which it ended up that way. And so that's one vehicle. All of our programs are open to businesses, and we've been funding businesses not only in the therapeutic area, but in research tools. We have tools and technology funding that we've done throughout the years too. So we have always been open for business, for industry, so to speak. What I've done in the last couple years wearing the GC hat is to look at some of our regulations that might not provide appropriate incentives, or actually maybe even created some disincentive for industry to join us. And what I've spent my first few years doing at CIRM is to amend some of those regulations. So I've been successful in setting forth some amended IP regulations that I think streamline it. Now, companies don't have to necessarily get loans. They can choose between a loan or a grant. So there's a lot of internal mechanisms that we've changed as well. In terms of going out and letting the industry know about who we are, I think they kind of generally know who we are within California, so that's good. What we've spent a lot of time in the last 6 to 9 months doing is meeting every single VC and pharma and showing them our huge portfolio of projects and trying to generate interest in their investments. And in the future, what I hope to do is also help the academics spin out these companies, or spin out into companies that can get funded through these VCs, because we have 77 projects that are early translation, and we'd certainly like to see a lot of those make them to the bedside.
So Brock, before you go, a quick question on that. So can you give the audience a sense of how much money has actually gone to a company through your greening process, and also what are the strings attached as part of that money?
Okay, well, the strings attached vary because they came in through different funding mechanisms. But for instance, we've recently announced funding for one organization that actually ended up with 2 disease teams. Our disease teams are $20 million a piece over 4 years, or up to $20 million. They usually ask for $20 million. So funny how that works. But we do a budget review after the fact, and so there is opportunity to set milestones and to also look at the budgets after the fact as well. So technically speaking, this organization has received up to $40 million. There's a few others that have received $20 million apiece. And we're excited about that. But there's others that are in our earlier stages, like early translation, and those tend to be up to $6 million a piece when they're just seeking to identify a candidate. Oh, strings attached is a 2-part question. Strings attached, all right, I mentioned loans and I mentioned IP, and I could talk probably an hour on all the specifics. It's really not that complicated. I'm trying to make it simple. So here's what we've done in terms of the grants, if you choose a grant, which I would think most of you would at this point in time. It is 0.1% per $1 million of net commercial revenue that's raised. So effectively, we share in your success, and you don't owe us if you fail. So I think that's it's a good way to make sure Californians get a return on their investment without burdening the companies that we're trying to facilitate. And the loan program, I could talk to you a little bit later about, that has warrants. At one point in time it didn't, so which is why it's a little hard question to answer, because if you came in earlier, you might have had what we call a multiple risk premium, and so some of those are a little bit different.
So we don't give money to companies, but we love getting money from companies, in all seriousness. One of the ways we leverage our investments is engaging in sponsored research projects with both disease foundations as well as companies. And one of the things we've found in our space is, in fact, the disease foundations are as eager and as motivated, in fact, drive a lot of the earlier stage research that then companies pick up. So for example, SMA was mentioned earlier today. Half a dozen years ago, very few companies are working in the SMA space, spinal muscular atrophy. We did some work in our therapeutic screening center that I mentioned, which is cell-based models of disease looking for drugs. So in vitro disease models, specifically looking for drugs, but it was funded by the Disease Foundation, SMA Foundation. Now, several large pharma companies have programs in SMA. So we like to partner on joint research projects. Several years ago, we did it was public, large 5-year, multi-year, multi-project deal with GSK, for example, and half a dozen different disease areas. And we have similar projects with other firms, many with disease foundations. One of the things that is true in this space is that, again, we use the term regenerative medicine broadly. The interest we've seen from the foundations and the companies is more on the small and large molecules, whether the drugs in and of themselves or the drugs to stimulate, again, endogenous repair, not on the cell therapies themselves, at least we have found in terms of interest. That's much more limited. On the outbound side, much as Elona mentioned, we work with our faculty in terms of licensing technologies, and we'll do that in a couple of ways. One is, the straightforward licensing will also help our faculty do startups, which again we're trying to help bring things out to the market. But we'll also do partnerships on the commercialization side. You may have seen a few months ago, there's an announcement that Janssen and Evotech did with some work coming out of our diabetes program, where Evotech, a small biotech, did a joint relationship with Harvard where cash didn't actually trade hands. But both parties committed to resources for a joint project plan. And then Janssen came along about a year later and said, we'll help take it to the next level. So there's sort of an interesting 3-way partnership between the academic institution, the biotech, and the big pharma company.
Can I just add, just because strings attached was brought up, we offer a number of programs in Maryland, and we don't have any strings. Just to put it out there, we only give grants. Those are not reimbursable grants. We don't take ownership of any of the IP. And we don't even force you to stay in Maryland if you don't like our environment. We think that you're going to love the environment, you're going to love the support. We have many other programs to leverage on funding at the later stages. And this is really the economic development hat that we have on, that we're not trying to reimburse the program. We know that this is taxpayer money from residents of Maryland to really support, to grow this life science industry.
Maybe as a follow-up question to that, Dan, Elona, can you speak to whether or not companies either outside of Maryland or outside of California are eligible, or how they might participate, or get some of the funding that you all are handing out?
So, for all of the programs that we have, you have to be a Maryland-based company. Exception is one new program that we're going to launch in about 2 weeks during the symposium, which is to do clinical trials in Maryland. And the idea would be that the company can be located anywhere in the US as long as they're going to open a site in one of the Maryland Institute or in one of the outpatient centers or in any location in Maryland. And then we can fund that specific site with up to $750,000. Again, with the idea that this would be a grant, not a payable, to take companies that already have clinical sites in many locations and they know that they need to open other locations, kind of encourage them to come to Maryland and to open a site in Maryland. This is the one that we have to offer to outside of Maryland companies.
Let me just start by saying what our statute requires. It's always a good place to start. Under our statute, Prop 71, which we talk about all the time, it says that we have to fund research in California. So therein is the focus. What's research? We do not consider research to be clinical trial because we think the research was the discovery of the candidate and then the creation of the clinical trial plan, which allows us to fund, first of all, clinical trials outside of California. Second of all, manufacturing is not research, we think, so therefore we can technically fund. But we do have another part of the statute that requires that 50% of services be, be purchased within California. So that as sort of the fundamental base gives us the latitude on an RFA by RFA basis to decide how wide the entry criteria would be. For instance, our first strategic partnership funding program was open to certain organizations meeting those key financial criteria that I just cited. Plus, they had to have at least 2 employees in California, and they had to have at least 1 clinical trial site in California. That's pretty liberal. I can't say that every single RFA would be like that. I know that California companies would probably like us to tighten that up. We have to wear many hats. We want to, as you do in Maryland, encourage clinical trials to come to California. We want to establish footholds in California. It's an economic development argument. Yet we want to also support the existing companies in California. So it's a trade-off about how stringent you have that criteria about the scope of ties to California. That said, we have always made sure that if there's a co-funding situation, the monies that we fund will be Californian.
Great, so I've got one more question and then we can open it up to the audience to begin to think about your questions. So you all have mentioned this field is early. It's in its infancy, so to speak. I think you made the argument that we need to have either government funding or foundation funding to move this along. Where do you see, each of your organizations going, let's say, in a 5-year horizon? And how different do you see it will be at that point in terms of focus, the way you fund, the way you think about this field?
So, first, I want to take a step back and say that I know it looks like maybe Maryland and California and Harvard are competing, and unless it's baseball, we're not competing.
We're not either.
And I know. Hopefully you're not going to compete tomorrow night.
Go Giants.
But actually one of our flagship programs that we have is collaboration with California researchers that we're very proud to have. As for where we are going, so we don't actually have we have a start date, we don't have an end date to our program. We were created as a Maryland statute that allows the governor and the legislators to fund us any year, depending on how much money they think that we need and how much money we're asking them, and we're continuing to ask them for increase, and they're continuing to argue that there is a need to fund other areas. However, we are part of a bigger plan that Maryland has. It's called Bio 2020. It's basically to infuse Maryland in the life science industry with about $1.3 billion until 2020. We are hopeful that we're going to continue to receive funding until 2020, and we think that we're going to be able to provide enough support to the legislators and the governor, whoever that might be, to continue to fund that sort of programs. We are keep on creating new programs. So a new program that's going to be, that just was created a year ago, is another infusion of other $6 million to translate the technologies that we already funded into commercial product. So we're keep on inventing other programs, and we'll keep on pitching those to the governor and the legislators, and very successfully to continue to support, in general, the life science, and in more particular, the stem cell and regenerative medicine.
Well, our situation is a little bit different, as I alluded to earlier. We were funded a fixed amount, and uniquely, though, we're not funded by the appropriations approach, which could be a future avenue. We were funded by something that's certainly, I think, from my perspective, more certain in that it's a bonded approach. So we were allotted $3 billion of bonds, and it was incorporated into the California Constitution, which means that we were not subjected to the whims of, you know, the politics and economics, which is good news right now in California. You know, we get our set amount, and it's there. It's very difficult to override what's been voted in years ago, and I credit Bob Klein with having the vision to structure this funding mechanism in that way. So $3 billion, and then we're done. I already alluded to when we think the last RFA would come out. It's probably 2016, 17. Then we have to manage them all. But that doesn't mean that we're done, because I think that, one, Bob Klein said that he would like to maybe seek another initiative for continued funding. I think that perhaps there could be the argument for an appropriations-based funding on a much smaller level, or maybe we are done. But even if this discontinues in California, I think that what we will have achieved and are well on our way to achieving is seeding the field so that all of these companies and these academics that have been coming to California and you know, accelerating the field in California will be garnering a lion's share of NIH funding, foundation funding, et cetera. So I think our primary mission was to help seed the field, and there's a possibility we could get continued funding, but if not, I think we will have succeeded in our mission.
The short answer to your question is I really don't know. So take this with a grain of salt. No, in all honesty, because, for example, when the Institute was formed, you know, 7 years ago, rough time for response now, for example, a lot of the energy was around making ES cell lines because there wasn't NIH money available for it, so it made them available to the field, great research. People weren't even talking about iPS cells and reprogramming in vitro disease models. Look at all the work that's happening now in specifically those fields. If that work pans out, I think one of the promises that this field holds is, can we fundamentally change the economics of drug discovery, which I think is exciting. So the next 5 years, I think, will show us whether we're right about going down that path or not. There's been a lot of talk over the years about, you know, bioengineering, three-dimensional tissues and scaffold, et cetera. And I think over the next 5 years, we're finally going to see, because we've learned a lot more about biology, that we're going to find the intersection between biology and, you know, material science, bioengineering clinicians, sort of paying off a lot more and actually yielding something, you know, substantive that will actually be relevant both for disease modeling as well as for therapeutics. Just look at all the work that's going on now in repopulating decellularized scaffolds and that sort of thing. Very exciting work that I think will have real clinical impact. So I see our move to the clinic, I see the basic, on the basic side, we'll, I think, find questions that we didn't even know to ask today. A lot of work happening right now in the intersection between genomics and stem cells, particularly at the single cell level, which I think is will be fascinating. And on the funding level, who knows? I said we are an organizational experiment. The good news is, we don't have a set termination date. The bad news is when we run out of raising money, we have a termination date. And from an organizational perspective, we may have said, okay, well, sort of we prove value, now maybe it should take a different organizational form. I don't know. So we'll have to see how that plays out.
Okay, so let's open it up for questions.
I'm probably the odd man out in this one. I'm actually a synthetic chemist trying to justify our existence. From all the observations I have seen and some wonderful work that has been developed in regenerative medicine, stem cells and all, an overwhelming amount of it has been directed to studying the fundamental biology of all these systems and then extrapolating it further. To many people, there is a little bit of a confusion about how exactly is this going to be a commercial product used to treat a very specific disease. In my humble opinion, not enough has been done to interrogate these stem cells and these biological systems with real small molecule drugs and chemical entities. What can be done to explore these kind of options? Because one obvious thing that I think of, and I have no experimental proof, but all this cord blood, all these stem cells, they are really extremely fertile territory for exploring drug metabolism. When I go to a clinic and I get a drug sample from a patient, they already are on 4 or 5 other drugs. So if you want to study some fundamental science, the cord blood and other things could be very useful, and also this can be extrapolated into using natural products and other things for regenerative medicine. Please tell me that I'm wrong, or if not, what can we do to change it?
One of the reasons we set up what we call our therapeutic screening center 5 years ago was specifically to do this. And let me just give you one brief example. I mentioned SMA before because that was one of the first projects we started with. So it's not, hematopoietic stem cells or whatever. But the whole idea was, how do you create in vitro disease models, interrogate that biology to find better drugs? So because we know that the SMN1 protein is downregulated in SMA patients, the idea was to create, because now you can do it at scale, whether you go from ES cell to motor neuron or IPS cell to a disease phenotype motor neuron. We can do that. We can do that at scale. And then you can interrogate it with compound libraries. And so the whole idea is, how do you find drugs that raise the level of SMN1 protein? With the idea that then you find a better, safer, more effective drug, because it's also been shown that drugs that have been tested so far, been tested in fibroblasts, et cetera, aren't effective when you get into patients. And so we're taking that model that we first sort of test in SMA into other disease areas, other cell types. Obviously, I think the field knows the most about motor neurons right now in terms of creating differentiated cell types, but the next step is to actually create differentiated, mature phenotypes of multiple cell types so you can do exactly that. And that's where I think the whole field of IPS and reprogramming are going to take us. And then it becomes even more interesting on the SMA as an example. The more we study it, the more you learn. So for example, people thought of that as a motor neuron disease. Well, we also find out, and there was a paper published a couple months ago where the man leading this, 2 of our scientists worked, one in muscle, one in motor neuron, looked at the interaction between muscle and neuron. And it looks like you may also think of it as a muscular disease as well. So looking at the neuromuscular junction, how does a muscle affect the nerve and the nerve affect the muscle? And then how do small or large molecules affect that? And so I think that's exactly what we're trying to do. And I think that's exactly what the pharmaceutical companies and disease foundations are interested in. What we have to know is when to stop. What we're good at is finding that first hit, finding the idea. What we're not good at is chemistry. So we shouldn't pretend we're good at chemistry. That's where we need the partnership, either with the CROs or the companies, to actually turn that into something that's clinically useful.
And we also have a $30 million effort at creating disease-in-a-dish models with iPS cells. So I'm sure that there will be a lot of small molecules that will be screened against those as well.
How do you assist in the translational process itself? I mean, recognizing that so many great discoveries come from academic laboratories, but academics are egregiously poor businessmen. And how do you structure that in your program so that you're helping people set up a company which is a reasonable idea?
Well, you certainly hit on an interesting problem. So what I found is that there's basically two worlds. There are actual PIs that are very interested in spinning out. So that those are the easy clients, so to speak. There are some that really aren't interested in spinning out, and I would love to explore more about Evotech because I think that is the solution. They want their programs to become more mature before they try to get VC or to deal with the pharmas, and I've got a few of those. And I think that this is a new model that I'll be talking to you about. And I met another organization that's interested in doing that type of work. So finding out about that model might be helpful. In addition, it's all about incentives and disincentives. So one of the incentives is, if you want to get funding for this program, you have to have VC or pharma funding or you don't qualify. So that's an incentive. We do not existingly have a disincentive that says you have to have a pharma or a VC. But at some point in time, we have always required that there be matching for clinical trials. So where is the financing for the matching going to come from? At some point, they have to start thinking that CIRM is not going to fund them all the way to phase through phase.
I guess the motivation, I think, is there. But how do you structure it? How do you help this professor with the idea to structure a company?
So one of the things that I mentioned is the Maryland Innovation Initiative. That's the new program that we have. It will bring business people with entrepreneur, with startup companies, with the inventor, with the tech transfer office, and we're going to provide the money, and we're going to provide all those people. That's part of the advantage of putting the Maryland Stem Cell Research Fund inside TEDCO. We've been around since 1998. That's what we're doing, translating the technologies from the universities to the commercial entities and providing the funding to do it. So we're hoping with other programs, not the stem cell program per se, but other programs that we have, that's what we're going to do. We're actually going to bring entrepreneurs to tour the labs and to find technology that they're interested in. Then we're going to give them money to take it with the inventor and to create startup companies around those.
And if I could add a couple of comments. We should also remember that not every good idea is worth turning into a company, and some should die in the lab, some should get licensed to a company, and some should turn into a company. In the course of if you go on the turning into a company stage, academic scientists can't take operating roles in companies. They can be scientific founders, they can be on the SAB, but they can't have operating roles. So to your point about they may be bad business people, that's fine. You don't want them running the business, and so sort of by rule they can't, unless they leave the academia. They work closely in partnership with the tech licensing offices, and two people next to you, you have Seema Basu from the Partners Technology Development Office. And also we have close relationships with not only the pharma companies, but also all the venture capitalists in town. And we're in constant conversation about when's the right stage, is there enough content here, what else has to be brought in, that sort of thing. So it's an ongoing conversation. And we help advise people on how to do that and what needs to be in it, how do you structure the IP, et cetera. So I think it's a combination of resources and then also being able to bring in people from the field. And I mentioned the Therapeutic Screening Center before. Because of that interface with the commercial sector, the person who's running it actually is an academic, but he had also worked in a biotech. So we have, some of our faculty have actually worked in companies or run science program within company, as well as worked in academia. And so, you actually have people with hybrid backgrounds in some cases who, are comfortable in both roles as well.
Okay, rather than ask a funding question, I'm going to ask for a prediction from all three of you. So, you guys kind of sit at a unique intersection, being able to see what's going on in both academic labs as well as in companies, working in the IPS space. How far away are we from solving some of the issues that have held up IPS, the epigenetic memory issues, the replicative senescent issues? From where you sit today, when do you expect to see the first IPS product in the clinic?
What do you mean by IPS product?
So just a cell therapy product derived from an IPS cell source. I'm hoping you're going to tell me we're a lot farther along than I think we are.
I think so, and I'm the non-scientist, so take that with a grain of salt. But from what I've seen, and I think we're a lot further than people might think.
I agree. I think the first step that we're going to see from IPS will be drug screening product, prediction of toxicity in different diseases, using different things, not so much in cell therapy. It's going to take a few more years. I don't know if the FDA actually approved yet even any product to even be considered. There is some interesting work in Maryland, Johns Hopkins done to reprogram blood cells using similar IPS technology that basically has no memory, and it looks very promising, and we're hoping that that might be maybe the first step, but we're a few years back.
And again, I don't know the answer to your question, but there are a few wrinkles that we have to solve. And don't forget, 5 years ago, we didn't know that with 3 factors you could turn a skin cell into an embryonic stem cell in essence, right? So who knows what's going to happen over the next few years. A few problems that still exist. One is, if you want to use IPS therapy for autologous therapy in the hospital, one of the issues you have is just how long does it take, right? Now, because of the reprogramming time, as well as, put aside all the epigenetic issues and all that sort of stuff that you were talking about, just the time to reprogram is an issue in terms of therapeutic relevance. So one of the questions, how can you accelerate that? On terms of IPS as a tool, we still have the issue of how do you create mature differentiated cell types from the IPS cell. So if you want to create, a line of, liver cells from someone with a particular category of disease, the field still doesn't know how to create mature hepatocytes, right? We have hepatocyte-like cells, but we don't have mature hepatocytes. So for example, one of the things I was talking about with some of our scientists about, could we create a consortium with pharma companies on, creating, in this case, we're looking at different neurons. We know the most about motor neurons, but we still don't know how to create a lot of mature neuronal phenotypes, whether it's dopaminergic neurons, pain neurons, all the ones that, drug companies would be interested in testing against. And you can't say you have a relevant model of disease until you can create an adult cell, right? And, what's it going to take to create that maturation barrier? I don't know. For example, in diabetes, people have been working on this for a while, just getting that last, maturation point of going from ESL to, insulin responsive beta cell still has to happen in vivo. And we don't know, right? I mean, we don't know what actually happened. We're trying to replicate that in vitro right now, but we still don't know how to do that. And people have been hitting their head against that wall for the last couple of years. So long-winded way of saying there are a few roadblocks before you can think of, I think, IPS as a real product, either as a cell therapy or as a tool. Another interesting thing I think to think about is also how to compete against what we call transdifferentiation or direct differentiation, where you could turn in vivo one differentiated cell type into another, and in fact not have to worry about IPS as a cell product. And that's just been explored literally over the last couple of years. It's been a whole new window that's opened up. So from a sort of competitive perspective, I think that may actually go faster than IPS and cell therapy product.
How we doing, Tom? Over?
We're done.
Oh, we're done. That's from the big boss over
If I
there.
had another people.
Well, I'd just like to thank the panelists. I mean, obviously, their organizations are doing a lot of work to really move this field forward. I think we all should thank them for their efforts and thank them for also the time on the panel.